Before we get into the process of protein synthesis, we first need to understand where protein synthesis takes place and what important structures help run the show. Let's head inside the nucleus, where we find our DNA. Our DNA provides us with instructions for making specific proteins. In this video, we will be demonstrating how to build a hemoglobin protein, also known as a transport protein. Hemoglobin proteins are responsible for transporting oxygen throughout the body. But before we can get to the building of protein, we need to transcribe our DNA. This process of transcription begins in the nucleus of our cells. The main goal of transcription is to take DNA and, using RNA polymerase, transcribe that piece of information into mRNA, also known as messenger RNA. Helicase then unzips the DNA molecule so you can have a template strand which RNA polymerase uses to transcribe complementary RNA, and a coding strand which does not get transcribed. To start off, we begin with the DNA template strand. Located on that strand are promoter regions that indicate where the transcription sequence needs to begin. A type of promoter regions that are TATA boxes, which is a genetic sequence located at the DNA template and is a recognition spot that the transcription factor proteins can bind to. Once transcription factors bind to the promoter region, it indicates to the RNA polymerase that transcription needs to begin. RNA then works in a 5' to 3' direction and binds complementary base pairs to the template strand, with uracil binding to adenine instead of T, and adenine to thymine, and cytosine to guanine. Now we have a piece of mRNA. As an added step of protection, an enzyme adds a 5' and a poly A tau, which is a stretch of RNA with an only adenine bases, to the 3' end to prevent degradation, which is destruction, when being sent out of the nucleus and into the cytoplasm. But before we get into translating, we need to edit our mRNA. Inside our genetic sequence are introns and exons. The exons are good and can be left in the sequence because they help code for our protein, but the introns that are non-coding need to be cut out in order to effectively build our hemoglobin. So to begin splicing our mRNA, we'll need a spliceosome. A spliceosome consists of several RNA proteins, and its job is to cut out the introns from the pre-mRNA molecule so that we have a mature mRNA ready to be transported out of the nucleus to be translated into the cytoplasm. After we have finished transcription, we head into the translation, the second process of protein synthesis. Our mature mRNA leaves the nucleus. It enters the cytoplasm where it meets the ribosome on the endoplasmic reticulum. The ribosome is our most important structure for protein synthesis. The ribosome, with the help of tRNA and aminoacyl tRNA synthetase, work together to decode the mRNA so that the right amino acids are put together and create our protein. The mRNA enters the ribose, composed of two subunits, a large and a small one that fit like a conform around the mRNA. Aminoacyl tRNA synthetase is the enzyme working to catalyze the attachment of amino acids onto the tRNA so tRNA can bring those amino acids to the growing polypeptide strand in translation. tRNA is the transfer molecule of RNA that is in charge of transporting the amino acids matching the anticodon, which is the sequence of 3 base pairs corresponding to the mRNA codons, onto the growing polypeptide strand that is translated during translation on the ribosome. Our first stage of translation is initiation. The ribosome settles around the mRNA template, and the transfer RNA brings over the amino acid, also known as methionine, that binds to starter codon AUG. mRNA is coded into triplets of bases, which are codons. tRNA needs to bring over the first amino acid to begin our translating. This completes our initiation complex. Next is elongation. During this stage, our amino acid chain is getting longer and elongated as the mRNA moves through the A, P, and E sites of the ribosome. First, the tRNA carries the anticodon with matching amino acid, methionine, located in the P site of the ribosome. This allows a fresh codon to be exposed in the A site, also known as the landing mark. Then, once tRNA lands in the A site with its next amino acid, the anticodon pair, we can connect the amino acids from the P and A sites to begin elongating our polypeptide chain. To understand why tRNA chooses a specific amino acid to bring, we need to look at a codon chart. A codon chart is read according to which codon is next to be moved through the A site of our ribosome. The type of amino acid depends on the pattern of letters in the codon. You first need to look at the beginning letter and match that to the left of the chart. Then you look at the top of the chart for the second letter in the sequence, and finally the third in the far right column. And you can connect each pathway down to find the amino acid associated with that codon. If we do start codon AUG, we begin at A, go up to U, and go down to G. So we know that the start codon associates with the amino acid methionine. Our last step is termination. Termination begins when a stop codon, such as UAA, enters the ribosome and triggers the release factors found in the P site to cause the enzymes to break the bonds and release the polypeptide chain so it can eventually leave the cell. Our hemoglobin protein is then moved from the endoplasmic reticulum, where our ribosome is located, inside a vesicle towards the Golgi, where it needs to be packaged. Then it is transported in another vesicle, also known as a fluid-filled sac, to be taken to the cell membrane, where it can leave the cell and begin transporting oxygen throughout the body. This completes our central dogma, our sequence of information processing beginning with DNA and ending with our constructed protein. Thank you for watching, and if you guys have any questions, please comment down below.